one of the reasons that Mike was invited, and he's very kindly making himself available from Australia, where it's a very inconvenient time zone for him, is that he had done some really interesting work, and he came to give a talk at the Sanger Institute, which I heard, talking about transcription in intergenic regions, and it was work that involved the catalogue curation work. And I see the catalogue slides on so many presentations, but I had never seen it before in this kind of presentation. And I was really excited about the work, and it's really great to have Mike presenting for us today. So Mike, we're really looking forward to hear your presentation.
Okay, thank you. All good to go then?
Good to go.
Okay, so thanks for the introduction. So as we said, I'm Mike Clark, and I split my time between the University of Queensland in Brisbane and also the Garvan Institute for Medical Research in Sydney. And I'm going to be talking about a project we have ongoing in the lab about uncovering hidden genes in intergenic GWAS regions. So I'm going to cover a couple of things in my talk. I'll talk a little bit about the fact that transcription is pervasive across the genome, and we'll sort of use that to ask the question, are there really going to be any intergenic genome-wide association study regions really when it comes down to it? I'll talk a little bit about a method we have for detecting transcription called RNA capture sequencing, and then I'll bring these two together to talk about how we're using this RNA capture sequencing method for novel gene discovery in these supposedly intergenic GWAS regions. So transcription is complex and pervasive across the genome, and this comes from the results of a number of studies. So if you can see this hypothetical coding gene I'm showing here in blue, when you have a locus in the genome, often what you'll have is your standard coding gene, and then you'll have a number of splice variants, coding variants around it. And then we tend to have a whole plethora of non-coding transcripts which surround and are within any coding gene loci. And so we get things like enhanced RNAs, we get RNAs from introns and things like slow RNAs, we get RNAs that come from transcriptional start sites and termination sites, and then on the antisense strand we get basically all the other things going the other way. And then out in sort of intergenic space, we also have the reasonably well-known now intergenic long non-coding RNAs, otherwise known as lincRNAs. And kind of the take-home message from all of this work, which came out of the ENCODE project last year, was that at least 75% of the human genome is transcribed, so it's fair to say quite a lot of this is transcribed at low levels. So transcripts can be both lowly and quite specifically expressed. And again, this is a slide I've taken from ENCODE. And if we look on the left here, we have a number of different tissues where we're looking at transcript expression. So in red, we've got a box on the top of coding genes, and in blue, we have long non-coding RNAs. And what's pretty clear from this is through all the different tissues is that some coding genes, but probably the majority of long non-coding RNAs, are all quite lowly expressed. And it's not just that. If you look on the right here, we have breadth of expression, which is looking at how often, what percentage of tissues is a transcript expressed in. And while the majority of coding genes are basically expressed in every tissue you look at, there are a few coding and a lot of the non-coding transcripts you'll find only in one or a few tissues. So you can have both low expression of some transcripts and then also very tissue-specific or tissue-restricted expression of some transcripts. And since we know that there is transcription across pretty much the whole genome, we get to the point where we ask, well, are supposedly intergenic GWAS regions really intergenic? And so I realized that we've already had a bit of introduction to GWAS regions, so I'll just explain how we've come up with sort of intergenic loci. So obviously, genome-wide association studies identify SNPs with a significant association to various traits or diseases. And 88% of these identified SNPs are intergenic or intronic. And given that coding sequences only make up about 3% of the genome, that's perhaps not surprising. But where it gets more interesting is when you start thinking about haplotype blocks or regions of linkage disequilibrium, because obviously the SNPs identified in these GWAS studies are generally not causative alleles. What they really are is a marker for a region of linkage disequilibrium around that SNP, that somewhere in that is the actual variant that's causing this trait or disease or contributing to. And what we find is that when we expand out from these SNPs, so we're including the whole haplotype block, we find that there are still hundreds of GWAS regions which are intergenic and don't have any known genes in them. And so our question is, are these really empty of genes, or is it simply that we've just not found them yet? And so the aim of our study is to try and discover novel genes within these intergenic GWAS regions. And the way we've been doing this is a modification of our standard RNA sequencing, but I thought for those who are not familiar with RNA sequencing, we just quickly outline it. So basically you have your transcriptome, which is made up of a lot of different transcripts. And to do RNA sequencing, basically you fragment the transcriptome into little pieces. You then reverse transcribe it to make cDNA, and then you sequence millions or hundreds of millions of your fragments, which you then align back to the genome. Hopefully you can see my mouse here where I'm just showing this. And then with a few nifty pieces of computer software, you then reconstruct this transcriptome, and then hopefully you'll be able to quantitate the expression of each transcript as it was originally present. There is a bit of an issue with RNA sequencing though, and the issue is that you're mostly resequencing highly expressed genes. And you can think of the transcriptome a bit like this pool of jelly beans here, and that it's quite complex and there's lots of different things. And RNA sequencing is a bit like eating some of these jelly beans at random. You have a pretty good chance of picking these common ones, but a really low chance of finding things which are quite rare or tissue restricted. And I'll just explain this a bit further in my next slide. So the problem with finding rare transcripts is that it's been found that the most abundant 1.5% of transcripts take up almost half of your sequencing reads that are done on the sequencer, while only 1% of reads are measuring or detecting the least abundant half of transcripts. So many transcripts, such as transcription factors and so on, and almost half the rest of it is only found by 1% of your sequencing reads. And then there's the issue of transcripts with restricted expression, and that's that some tissues are very heterogeneous, and therefore if you have a transcript expressed in only a small minority of cells, it becomes very hard to find. And I've included a bit of an example here that if you have a transcript expressed at an average level, say 10 copies per cell, in 0.1% of brain cells, which actually is around about 170 million cells, so a lot of cells, you would need at least 400 million sequencing reads to have at least one read or one piece of sequenced RNA to cover that whole transcript. The problem is that you really need much more depth than that to properly detect and quantify transcripts. So you can see that when you have a heterogeneous tissue, you have basically an almost insurmountable task to see things which are restricted, even though they may not be rare where they are expressed. And so the question becomes, how do we actually find these rare or restricted transcripts, which we think are likely present in intergenic GWAS regions? And we've been using a technique called RNA capture sequencing because it provides a much better way of detecting these transcripts. So RNA capture sequencing is a fairly novel method. There's only a couple of papers about it, so I'll just give a quick rundown. Normally with RNAseq, what you're essentially doing is sequencing the whole transcriptome. With CaptureSeq, what we do is we select a portion of the transcriptome, and then we do focused sequencing on that only. And what we're able to do there is achieve a huge increase in sequencing depth and coverage of what we're interested in. So if you have your RNA transcripts here, which are turned into cDNA, and these darker blue ones are the ones that you actually want to sequence, what we do is we design oligonucleotides against either exons or transcripts, or even just tile along regions of the genome where we want to find and pull out and then sequence any transcripts that exist there. And so we have our oligonucleotide probes. They hybridize to what we're interested in. We then pull them down, wash away everything we don't want, and sequence the things we do. And then this approach provides a great benefit when you're trying to look for rare or transient or tissue-restricted transcripts which are below the detection limits of most conventional RNA sequencing. And so we published an example of this a couple of years ago. So this here is a Surcos plot, and each of the different colors which I'm showing with the mouse, hopefully you can see, represent a different, basically gene desert. And in green you can probably see what I'm showing up the mouse here, these were known, say, splice functions, so known transcription through this region. And you can see that some of these areas had no known transcription whatsoever. And this was what you got with normal RNA sequencing. On the other hand, when we captured these regions and then sequenced, we got a huge increase in transcription through this region. And so in blue now shows all the exon junctions we can now detect. And so you can see that where we previously could see no evidence of transcription, we can now see quite complex splice transcription. I should mention this work was done in collaboration with John Lynn at the Broad and also Roche. So therefore we thought, well, we can use our capture sequencing to look for novel genes within these intergenic GWAS regions. So what we did was we took ribodepleted RNA from 20 normal human tissues. We designed oligonucleotide probes that targeted basically the whole haplotype blocks we were interested in, and we had 339 of them. And then we had probes tiling across the whole region, and then we pulled down any transcription that came from these regions and sequenced them. And part of the reason we did this is that an advantage of looking at these GWAS regions over, say, just an average gene desert or in other intergenic regions is we know these regions are functionally relevant, so we think that the transcripts we'll find have a higher chance of being functionally relevant. And so these 340-odd regions targeted about 150 different traits and diseases identified by GWAS studies, so everything from alcoholism to hip-waist ratio, so quite a variety of things there. And we were targeting about 50 megabases of the genome. So what did we find? Well, the first thing you want to do when you do this capture sequencing is check that you really have got a very good enrichment. And so an easy way of doing that is to do standard RNA sequencing, then use the same RNA, capture the stuff out that you want, and then sequence that, and then say what percentage of the transcription that we see came from the regions we were interested in. And so I'm showing this in this graph here, and we can see that pre-capture, these GWAS regions or transcription from these GWAS regions accounted for about 0.2% of our library, but after capture, it accounted for 60% of all the sequencing we had. So we had about a 336-fold enrichment of transcription from these GWAS regions in our sequencing library. This obviously allows us much greater depth to look at what's there. I'm just showing here like a control loci that we had to sort of show what actually happened. So in red here, I hope you can see the mouse, are the regions we were capturing. So basically all the exons of this housekeeping gene, HMBS. And then this is from UCSC Genome Browser, is basically showing how much sequencing reads or sequencing we had from each of these captured regions. And in blue here is after capture, and in green here is pre-capture. And I've put these two tracks here on the same scale. So you can see that post-capture we had enormous sequencing depth over these exons, compared to pre-capture. And then down here in blue and green are the transcripts that we were able to construct after our capture sequencing. And so you can see that we do get reasonable looking transcripts, and we also actually, even within a housekeeping gene, start to see what looks like novel isoforms. But what about these GWAS regions? So we see quite extensive transcription within these supposedly intergenic GWAS regions. 85% of the 339 regions we looked at contained splice transcripts, and the graph here on the right shows the fraction of regions which had different numbers of transcripts. So you can see that a small number had 0, but some even had as many as 20. We see a median of 4 splice transcripts per loci, and we also find, quite interestingly, although perhaps not unexpectedly, that this transcription doesn't just stay within these GWAS regions, that many transcripts are transcribed either into or out of the regions we were capturing. And so we were able to identify around 1,500 transcripts in these intergenic GWAS regions. And this is post-capture, that compared to only about 100 pre-capture. The majority of transcripts we found were either entirely novel or new isoforms of ENCODE genes, so I'll just go through this. So about 40% of the transcripts we found were new isoforms of ENCODE genes, This generally means that what we've found is perhaps a novel 3' or 5' extension of these genes, which extends these gene models into these GWAS regions. About 60% of the transcription we found was completely novel and never been seen before, and these varied a bit between sort of intergenic or antisense or intronic transcripts. And a very small percentage turned out to be known, and that's probably because in the time it took us to sort of design and then carry out the study, a few of these GWAS regions had transcripts annotated within them by gene code. A high focus of our lab over a long time has been to look at long encoding RNAs. So we were interested in how many of these novel transcripts were linked RNAs. And with some pretty stringent filtering, we identified that at least 650 of these new transcripts were LNC-RNAs or linked RNAs. And all of these were either completely novel or there was about 30% were new isoforms of gene code genes. And so none of them turned out to be actually known. And then you might say, well, so we had 1,500 transcripts in total and about 650 were non-coding RNAs. What were the rest? And so the rest are generally novel isoforms of coding genes. And then there's also, which is something sort of defined by John Rin's lab, transcripts of uncertain coding potential. And these are transcripts where it's very difficult to say one way or the other whether they encode a protein. So I'm going to show a few examples of some of the transcription we find within some of our GWAS regions. So this is here a human loci that's associated with HART-QT length. It doesn't contain any known genes. HART-QT length is obviously important because if you have abnormal QT length, it predisposes you to some cardiac arrest. So this is also from the UCSC genome browser. So in red here we have some of the GWAS SNPs that are found in this region. In sort of a yellowish color or yellowy orange we have the haplotype block through this region. GenCode has no annotations in this region. Our pre-capture sequencing identified no transcripts in this region. A couple of years ago, John Rin's lab used the Illumina Body Atlas to publish a large set of intergenic long encoding RNAs. And so that was, sequencing of about 16 tissues to quite high depth. And they had, shown here in green, found one little free exon transcript. But after our capture sequencing, we're able to see that this transcript actually just forms perhaps the nucleus of a much larger transcriptional network that's found through this loci. And so we find many new transcripts here. And obviously the good thing now is that if you were interested in this GWAS region, you now have some transcripts which you can investigate and test for their functionality and whether they have any impact on the association of this region with QT length. I mentioned before that some of our regions we have novel extensions of known genes, so I'm just going to show an example of this. So this is another region. There's a couple of GWAS SNPs that are associated with it. The haplotype block is shown here. A little bit upstream is a known coding gene called EMBL-EBI. We don't find any transcripts in our pre-capture sequencing, but post-capture we find several transcripts, including this transcript here, which I'm just showing with the mouse, and that has a novel 3' extension of this coding gene that contains essentially, although not completely, all of the normal coding region. On the other strand, we find a number of transcripts, and these are heading from left to right. And we don't really know a lot about them, although we do know that very recently a microRNA or putative microRNA was annotated within this GWAS region on that strand. So it's possible that these transcripts here are the host transcripts of this putative microRNA. Along with discovering completely new transcripts, one thing we've found about CaptureSet is it's very good for resolving and improving the somewhat fragmented annotations we sometimes have around the genome. And so what I'm showing here, you can see in red there are 3 large haplotype blocks, and they're all associated with prostate cancer by quite a few studies, and this is a region on chromosome 8. And you can see in green the lines that represent the number of GWAS SNPs that have been found throughout this region. On the right, if we look down at the gen code annotations in green, we can see that the right couple of haplotype blocks do actually now have some transcripts annotated within them. And on the left, we have some quite small sort of 2, 3, 4 exon transcripts annotated both by Kibili and John's RIMS group and also by gen code. And when we do our capture sequencing, what we find is that these are sort of generally sort of fragments or pieces of what appears to be a very complex non-coding RNA loci with a lot of alternative splicing. And so that's definitely one advantage of what we're doing, is that we're able to sort of take fragments that, what my guess is that with RNA sequencing they were too low to be properly detected and therefore assembled, but with the extra depth we get from capture sequencing, we can now start to resolve the transcription within these loci. Just to point out that we are quite happy with the kind of depth and assembly that we get. There was very recently a newer IncRNA catalog published, and so I'm showing their IncRNAs in black up here. And so this involved about annotating IncRNAs from about 125 RNAseq libraries. So there's a huge amount of standard RNAseq data. But what I think is clear is that using capture sequencing and basically sequencing one or a small number of libraries, which we show here in blue, probably gives a better result for annotating low-level and detecting low-level transcription. So just in summary, with RNA capture sequencing, we're able to get unprecedented depth of coverage and enrich and identify transcripts that you essentially can't identify by standard RNAseq. We find extensive transcription both within and across what had previously been called Intergenic GWAS regions, but probably turns out they're actually genic. And we identify 1,500 mostly novel transcripts. These novel transcripts provide our candidates to explain the traits of at least some of these regions, or at least allow people to now start asking questions about these regions and what they do, armed with the information that there is genes or transcripts within them. And because, again, we're people who like to look at non-coding RNAs, we think that using these Intergenic GWAS regions with CaptureSet is a good method for identifying novel and potentially functional lincRNAs compared to just identifying them generally throughout the genome. So all that remains for me to do is thank the people that helped me with this work. So at the Garvan Institute in Sydney is John Mackett and Marcel Dinger, and also Tim Mercer, who's helped work with me on the informatic analysis. And up here in Brisbane at the Institute for Molecular Bioscience, I should thank Joe Crawford, who's worked with me closely on all of the lab-based work. And then have to thank Jeff Gedlow at Roche and Daniel Gerhardt, who's also in Brisbane at the Major Medical Research Institute, for all their technical support. And thanks everyone for listening.
Great, Mike. Thank you very much. If you could unshare your application, we will be able to see any questions that come out of the chat window, because when you're presenting it disappears. So that would be great. And then in the meantime, we could also take any questions from anyone who is dialed in.
So as a reminder, if you wish to ask a question by the phone lines, please press *1.
That was fantastic. A really, really interesting set of observations. I wonder if you could say, of the regions that you started out with as looking like gene deserts, what proportion of those now end up with still being gene deserts?
So about 15%. Although I think it's worth noting that if we looked in more tissues, some of those probably start to be genic. And we mostly limited this analysis to splice transcripts because they're much easier to differentiate from perhaps any background. And so I think also when we continue the analysis and start to bring in perhaps more single exon transcripts, I think the number of supposedly still gene deserts will decrease again.
So, Mike, I had a question about how we actually should present work to the community. So clearly we have a lot of experimental evidence, sometimes from multiple sources, sometimes from an annotation project like ENCODE, and all of these things are displayed in genome browsers. But as his information gets very dense for a user who's coming in looking at variant level, and it's kind of hard to think about how a user might want to actually interact with that information. So how would you prioritize it and how would you display it?
It's quite a tough question because I guess that, long experience makes people like me quite comfortable with the browser. I'm not really sure how you can take what now must be thousands of data tracks and make it very easy for people to kind of find the ones that are important to them. I might have to pass on that for the moment. You might have to get back to me in a couple of days.
Okay. And then in terms of sort of showing users prioritize data, would you, for example, restrict data that you displayed to items that were described by multiple data sets, where you could actually sort of see a sort of best of breed this is confirmed by multiple methods?
So it wouldn't be a bad thing to be able to display things that have been shown repeatedly, although I suppose to some degree that's kind of what GeneCode are trying to do, is they're trying to take things that seem quite robust in their annotation, or things that really do seem to be there, and make a catalog of them. I think one thing that would be really great is that if you could look at one of these genome browsers to see not just the transcripts that have been found, but also kind of the tissue and expression level information about them. So I think that would be very useful. And just to go back to the finding them multiple times, if you take sort of our sort of data, because a lot of this hasn't really been done before, you probably find that compared to all the other data sets, a lot of the stuff we find hasn't been found before, and therefore it may be just as reliable. It's just that we're using a more sensitive method. And so if you only ask for things that have been seen multiple times, you might, for example, not find our data.
Okay, thank you. Any more questions for Mike?
I see that I've got a Q&A question.
Okay, great.
The question is, what are the possible roles for these long encoding RNAs? And I guess they would be the same sorts of roles that we find for existing functionally characterized long encoding RNAs, which are actually very various. Generally speaking, they regulate gene expression, but they do it at multiple different levels. So some, for example, bind to chromatin modification complexes and therefore silence or activate other loci, and they do this both in cis and in trans, depending on the RNA you're looking at. There's an emerging role for other long encoding RNAs to go into the cytoplasm and act as microRNA sponges, and therefore regulate gene expression. Some seem to regulate splicing or nuclear export or say chromosomal structure or nuclear domain structure. So the roles are basically as varied as you probably could possibly be. I hope that answers your question.
Okay, thank you, Mike.